Hello everyone. So we will let everyone enter the room, and I would like to say hello. So my name is Clémentine Schild. I'm the head of digital education at Institut Pasteur, and I would like to welcome you on this live event of the MOOC Human Population and Evolutionary Genetics. And I would like to thank our director Etienne PATIN and our community manager Oguzhan PARASAYAN for creating and animating this online course and joining this live event. Thank you very much. I will encourage you to present yourself in the discussion, as I will do it in shortly. So indicate your name and where you're from, and also what you're doing. And then for your question, it's much easier if you use the button called Q&A that is on the right side. So you should see a button Q&A. So please use this one for your question. And if you have technical question, don't hesitate to use the discussion, the chat. Speak slowly, weak connection here. Sorry for you. Well, we
Maybe
will try.
Just to complement, you can ask questions in French. We will translate it to English.
Thank you, Etienne. And Oguzhan will also start with questions that were asked in advance.
Yep.
So for now, please present yourself in the chat.
Yeah.
While okay, I think Oguzhan, you can start, and people will continue to present themselves in the chat.
Okay, perfect. Thank you, Clémentine, for the introduction. First of all, we hope you enjoyed the course so far, and we are all happy to be here all together with you in this live session of Human Evolutionary Population Genetics MOOC. If I start to introduce myself, as Clémentine did, I'm Oguzhan PARASAYAN. I am a postdoctoral researcher in the Human Evolutionary Genetics Lab at the Institut Pasteur, and I'm working on briefly demographic and genetic history of human populations using both modern and ancient genomes. And I'll let Etienne to introduce himself.
Thank you, Oguzhan. So hello, everyone. Thanks for being here. That's a real pleasure to be here with you today. My name is Etienne Patin. I'm a CNRS researcher in France at Institut Pasteur. I'm a population geneticist working on a number of different topics, all related to human evolution and history.
Thank you, Etienne. So far, we will try to answer your questions that we will receive in question Q&A section. But if we don't have enough time, in the end, we will continue answering them in the discussion. So we had so far one question in the discussion part of the form, which was are there any genetic studies on Indian populations, and are these metabolic? Sorry, because I'm translating in from French. Do we have the same metabolic changes in the Tibetan that we see in the Tibetan populations as in the Indian population? So, what would you like to say, Etienne, about this?
So, what do we know about Indian populations? So to make a long story short, we think there are more or less three populations that contributed, ancestral population that contributed to the current gene pool of Indian populations. One of them is probably related to the first inhabitants of India who migrated from like the northern part of the Arabian part to the South Indian Peninsula a long time ago, probably like 40 to 50,000 years ago. And so these populations are not observed today in the way, obviously, they were at that time. But we tend to think that populations from the Andamanese Islands, so the small islands that are close to India, are some kind of a close group to the populations that arrived there initially. Obviously, they changed since that time, right? And so then we see this genetic ancestry related to the Andamanese more common in the south of India compared to the north. And so what do we observe in the north? So it's another genetic component, another ancestry that is related to migrations that occurred at the end of the Neolithic and the beginning of the Bronze Age in Europe, but the same occurred also in India, and it's related to the Yamnaya populations from the Caucasus. So there is today a gradient of these two different components that we see in India, a gradient that goes from north to south with more Yamnaya-related ancestry on the north and more Dravidian ancestry in the south. You also have other ancestries related to these ones, and one of them, the third one, is actually related to Northeast India. I see a question related to that. So populations speaking Austroasiatic languages. And this one is observed in few populations with specific languages that are part of the Austroasiatic group and are not very well studied, actually. So that's a very general idea. And so about adaptation to high altitude in Indian populations, I don't think we see any clear, but maybe I'm wrong. So just to make a quick introduction to this question. So we know, based on the comparison of the genomes of Tibetans and Chinese populations, that one or two genes have participated to the genetic adaptation of Tibetans to high altitude, because they show very strong genetic differences between these two groups, while the two groups are relatively and another one, 𝐏𝐚𝐬𝐭2 and 1. Sorry, I see my connection is unstable. Could you hear me?
Right now we can hear you, yeah, but there was a one.
Okay, sorry. So two genes, including APAS1, so probably participated to the adaptation of Tibetans to high altitude. I'm not going to get into the details of why and how, but it seems like these mutations are not particularly observed in most Indian populations. But again, there are actually few studies focusing on, for example, Nepalese populations and populations that are on the southern part of the Himalaya. So there may be surprises there.
Okay, then thank you, Etienne.
Do you want to complement, Oguzhan?
Also the question was about like Andean, sorry, how to pronounce Andean populations from South America as well. If we could also add something about that, their adaptation to high altitude. We see that they have the similar phenotypic adaptation to the high altitude, but there are many studies are like done on these populations, and the overall results are not like fully shared. Shortly, we see that the genetic background of this adaptation is different than Tibetan populations, and we can just conclude that it seems like a convergent example of a convergent evolution, the adaptation to high altitude. So different populations adapt in a with different genetic mechanisms to high altitude.
Yeah, they converge to the same phenotype, being adapted to high altitude through different biological mechanisms, different genes and different mutations. Yeah, this is convergence. Yeah. So yeah, there is genetic evidence for adaptation in the Andes, but different genes basically than the ones found in Tibetans, and it's the same actually in high altitude Ethiopia.
Thank you, Etienne. And if we continue with the questions, first question we received in the during the registration to Zoom, the live session. Native American populations were the last to migrate, so their gene pool would presumably be smaller. Has this been observed? And if so, would this impact their ability to adapt to the environment and develop diseases?
That's a nice question. So again, I need to introduce a number of quick concepts to understand the question. So the first is, as said in the question, probably South America was one of the last, that was probably the last continent to be peopled by modern humans. Although we need to keep in mind that Oceania and the most remote part of Oceania was peopled even more recently, like 3,000 years ago. So it is assumed that the peopling of the world by Homo sapiens was done through a series of founder effects, founder events. What does it mean? It means that as Homo sapiens left Africa to people the rest of non-African continents, only small groups left the previous population. So at each migration, only a subset of the genetic diversity was kept and was migrated with the individuals. So it's like at each step in this long process of peopling in the entire planet, only a subset of the genetic diversity was kept. Like you take some balls in a bag and only a few balls in this bag of diversity. And this process makes that the further we are from Africa, the lower the genetic diversity. And so this is indeed observed in South American populations. We see lower genetic diversity in Native American populations, particularly if we look at population isolates in the Amazonia forest. It's not exactly the case for the rest. I'm going to come back to that quickly. And so what we see in these populations is typically low genetic diversity within populations, and also relatively strong genetic differences between populations. Even if these populations are connected together and they separated recently from each other, they show a lot of genetic differences. And this is due to genetic drift, that a concept that was introduced in the course. So just to continue with Native Americans, we need to keep in mind also that the peopling history and the gene pool, of South Americans was completely changed by European colonization. So obviously the genetic diversity today for most of these populations has been also affected by this colonization event. And so there is a lot of European and African, sub-Saharan African ancestry today in the Americas. Regarding adaptation, so that's also an important question. The idea is that if you have lower genetic diversity, you have less mutations to adapt to your environment. It's even, we can actually reason that this lower adaptation is also due to the fact that you have more genetic drift in that population. So very quickly, you know, based on the courses, that genetic drift is the mechanism through which mutations fluctuate a lot in frequency from one generation to the other and is disconnected from natural selection. It can actually prevent selection to be effective. So deleterious mutations can increase in frequency under genetic drift. Advantageous mutations can decrease in frequency under genetic drift, and the other way around. It's like a stochastic process in small populations that make allele frequencies fluctuate a lot. So just to come back on the question, so in Native Americans, because the population size is relatively small, plus the genetic diversity is also not that great, we could imagine that they have lower capabilities to adapt to, for example, the pathogens that we can find in the Amazonian rainforest. So the short answer is that's not the case for two reasons. One is just empirical evidence. We do see evidence of genetic adaptation to some, for example, pathogens in Native American populations from South America, and for endemic pathogens in particular. So we have empirical evidence for this. And the second is that we actually see no human populations where we have high frequency deleted mutations that will really increase the prevalence of diseases in these groups, like very clearly. And the reason for this is that these founder events that I mentioned are too recent or too weak to really affect the genetic diversity very strongly. So to see these kind of effects, you need population size reductions that are much stronger or much older than the ones we see for Native Americans. Long answer, but everything is said.
Thank you, Etienne. I think I don't have anything to add. It was a developed answer. And another question we have received during the registration is, can you explain how the genetic influence of Yamnaya people or Yamnaya people from Pontic steppe is reflected in the genes of Europeans? Are there specific characteristics that we have inherited? Did they bring with them any Denisovan genes? I can start a bit and
Go
then
ahead,
maybe
Oguzhan.
you can add up. So the thing is, the first thing we see in terms when we discuss this steppe ancestry or like this famous steppe ancestry in Europe, it's in terms of ancestry. We in all Europe, present-day Europeans, we have the ancestry, this steppe ancestry that initially found in the people associated with Yamna culture in the Pontic steppes. And there is as Etienne mentioned already earlier, there is what we see is after the various migration and admixture events, there is a gradient of Yamna ancestry that is found in the present-day Europeans. That is something prehistorical or historical because of the historical events. Like in the north or northeast, we have tendency to see higher Yamna ancestry, which degrades in the more southern or southwestern part of Europe. And if we look at the specific characteristics that are inherited, this is not an easy question to answer. There are studies going on, but for this is, I think we can call it fine mapping, right? It's like to find the specific variants characteristic in the ancestry specific characteristics. For this, we really need a high quality of and high number of Yamnaya and like genomes that represent the steppe ancestry. However, this is not the case for the moment. Like there are a few high quality genomes, and there are a lot of genomes around done with capture method, which is not a like whole genome sequencing. So, however, there are still some studies worked on this. We know some pigmentation genes like light skin pigmentation were arrived or was already fixed with in Yamnaya people or steppe ancestry people. So this increased this frequency of this allele in European populations. In some studies, they showed the tall stature was correlated with the Yamnaya ancestry. And in one recent study also showed like there might be a connection to high cholesterol with the Yamnaya ancestry. But I think this is a question really to be answered in the future, because for the moment we don't have enough power. But I think in the future we will recent future we will answer this question in more detail. And to finish the last question, did they bring with them any Denisovan genes? No, I could say no to this question because there's no direct connection with Denisovan and Yamnayas. We find Denisovan ancestry today in the more eastern like East Asia or Southeastern Asia, as well as like Oceania. But Yamnaya population did not have Denisovan ancestry, and they did not bring any Denisovan ancestry to the Europeans. You don't find any Denisovan ancestry in European populations, for example. And I let Etienne to continue.
I have nothing else to add, except that you also have a bit of Denisovan ancestry in South Indians. This is usually overlooked, and it's probably related to early migrations, the ones I mentioned just before, not Yamnaya ones, right? So that would suggest that 𝐏𝐚𝐬𝐭𝐞𝐮𝐫 really have not brought additional Denisovan ancestry compared to what was already there in Europe.
Thank you. And the questions that we received during the registration are done for the moment. So if we go back to start with the Q&A, I think the first question was can you throw some light on the population genetic history of Northeast India? Etienne already did, but I don't know if you want to have something more to add.
Yeah, I would just say that we definitely need studies from that region, which is not well characterized. There is actually one very burning question that I already mentioned before regarding the origins of people from this region. And it's related again to these Austro-Asiatic languages. We don't know where these languages originated, and they actually are spoken in India, in Nepal, and also in Southeast Asia, with no link between the two regions. It's like two pockets of languages that are spoken far away from each other, and we don't know where is the origins of these languages and the people who speak these languages. We don't know if the migration was from Northeast India to Southeast Asia or the other way around. So it would be really cool to have new studies on that region, really.
I agree. We continue. Next question. Could you tell me more about the sex bias in North American from Asian genome? For me, it's not so clear, the question.
I'm not sure. Could you elaborate, Pierre? Maybe, just to reformulate maybe a bit.
Then we can
sex bias in North America, Asian genome.
There is another question, but okay, it's a different question.
Unrelated one.
Yeah.
Yep, I'm
So
not sure.
While waiting another question, I think maybe we can continue with the questions from Sergio. Could we talk about the Portuguese being a distinct ethnic group due to having unique genetic makeup in the form of high frequency of HLA haplotype? The Portuguese have a unique characteristic among world populations, a high frequency of HLA A25, B18, DR15, and A26, B38, DR13, and being the only one displaying the displaying the HLA A25, B18, DR15. I wasn't, yeah.
Yeah, that's a very precise question,
Yeah.
Sergio. Thank you. I have a general answer to that. First, the HLA locus. So the HLA locus is a specific locus. We need to keep in mind that this is actually the place in the genome that is the most diverse. So it's like any mutation occurring in this locus will increase in frequency. The cause for that is complex, but very briefly, the HLA is expected to be important in the immune response to pathogens, and there is some kind of an advantage for diversity. So what we call in general frequency-dependent selection. So it's like any rare mutation would be advantageous and will increase in the population. That means that these haplotypes that you mentioned, Sergio, are probably not only affected by demography and genetic drift, but also by selection. And that's something to keep in mind because we should be very careful, like raising conclusions from HLA about population demography for the Portuguese, for example, while we know that the same markers, genetic markers we are talking about are affected by natural selection. So it could be that for whatever reason, the ancestral populations that form the Portuguese populations today have been exposed to specific pathogens, for example, that would make these haplotypes increase in frequency. And that doesn't necessarily reflect the genetic difference we see between Portuguese populations and other European populations, for example. But in any case, there will be slight differences between Portuguese populations and others that you can always detect, but we need to keep in mind that this is not like one mutation that is very different between Portuguese and Spanish people, for example, but a lot of mutations that are very slightly different between the two groups, but they are sufficient to separate the two populations. Be careful with these. This separation is not correct in the sense that genetic diversity is continuous in this region and in many regions, so that actually there is really like a continuum of individuals between the most southern part, for example, of Portugal and the southern part of Spain, for example. So this is continuous. We want to classify these groups. We could try, but there will be individuals in the middle that we will have hard time to classify. And second, yeah, keep in mind that most of these genetic differences that we could observe between populations are usually non-coding. They do nothing on the phenotypes. And so we should be careful with these in general. Yeah.
Etienne, we had a modification on the reformulation of the question from Sergio, but I think your answer would be, I think, in the same line. Just to read again Sergio's question, the reformulated version of his question. Could we talk about the Portuguese being a distinct ethnic group due to having a unique genetic makeup in the form of high frequency of HLA haplotypes? And so this was
yeah. So I reiterate my answer. HLA is not representative of the rest of the genome. So if you see genetic differences between groups based on HLA, doesn't mean these populations are largely different. It could be simply that there has been local genetic adaptation to a specific environment there that makes the specific locus, HLA, more different between populations than what you could observe for the rest of the genome.
And we received from Pierre another version, the first question, like he updated the question. So in a video, you speak about a bias from female having more mutation from Asian population than the male in North American population. What is the order of this difference? Any hypothesis about this?
Let me read it again. Sorry, yeah. Are we speaking about we're speaking about North American populations so more mutations. Yeah, I'm not sure. Pierre, do you remember? Because usually when we observe sex bias admixture, it's a case where an unmixed population derives, for example, from two sources. Let's imagine here that North American populations derive from Asian populations and I don't know, like European populations. I don't know exactly what is the event you refer to here, Pierre. So sometimes this admixture process between two groups is sex biased, meaning that individuals, women from the Asian population tend to marry with men from the other population. You would observe in the genomes of present-day North Americans deviations not on the 22 autosomes, so these pairs of chromosomes that we inherit both from our father and our mother, but we will see that for these chromosomes that are sexual, we say, so the X chromosome, the Y chromosome, and the mtDNA. So these three different chromosomes are inherited differently. You know that already probably, but just to remind you, so Y chromosome is inherited from father to son only, because it determines the fact that an individual is a male. The mtDNA is transmitted from the mother to the kids because it's transmitted with the cytoplasm of the oocyte, and from mother to kids, and then among these kids there are men that will not transmit this mtDNA. So at the end, it's transmitted from mother to her daughter, and then this is a representation of the maternal lineage of the population. And the X chromosome, as you know, women have 2, men have 1. So when you have 2, when you tend to transmit more chromosomes from the women than the men over generations, because women transmit both, while men transmit just 1. So because of these different transmissions that are sex biased, we could see different contributions of the parental sources. And that's an interesting way to analyze the genetic data to learn more about how was the admixture event at the origin of a population. So basically, if we take examples of, I'm going to take example of African Americans because I'm not sure about Pierre's example. African Americans are today, on average, like 80% of sub-Saharan African ancestry, 20% of European ancestry. On average, again, it varies quite a lot within the population. So if we look at the autosomes, we see these 80-20% proportions. If we look at the X, so why is it different from the autosomes? Could you still hear me? Yeah.
Yeah, we can hear
Okay.
you.
Probably a mixture between African slaves and European settlers was biased towards intermarriages between European males and African women. So because of this, you tend to have more African ancestry on the X chromosome compared to the autosomes. That's like one way to learn more about how this admixture operated in the past using the genetic data. So sorry, Pierre, because I'm not sure what you meant exactly with these North American and East Asian population. I'm not sure which one you refer to.
Afterwards, we could also continue exchanging the discussion part of the forum, and like we can also find which video was it, and then explain more in detail in later on. Thank you, Etienne, for answering this question. So another question from Pierre is, in the site frequency spectrum, how can we distinguish a stable population from an increasing population or expanding population? What is the limit for just single mutation bar? Is there some equations or model to make model? Could you provide reference on this topic?
Yes, Pierre. So we're going to provide detailed answers with references because that's a long, that's a complex question. So, but to make it short, have very well described summary statistics that we could use to test if a population is under expansion or not. And the most used one is 𝐏𝐚𝐬𝐿. 𝐏𝐚𝐬𝐿 is basically measuring departures from the expected site frequency spectrum under a constant population size model. Under this constant population size model, which directly derives from the coalescent theory that I'm not getting into today, you expect that the first bar on the plot is around 25 to 30% of what we call singletons, mutations that are observed in just one individual in the sample. While under expansion, you would observe like 40% or more. And 𝐏𝐚𝐬𝐿 is basically developed to detect that. If you have an excess of low frequency mutations of singletons, 𝐏𝐚𝐬𝐿 is going to deviate from 0 and gets lower value, lower than 0, so negative values. And you could test, we know how the null distribution of 𝐏𝐚𝐬𝐿 look like under a constant population size model. And so you could see, yes, my 𝐏𝐚𝐬𝐿 is like very negative, and it's like outside of the distribution I expect under constant population size. So I conclude there is an expansion. That is something we can definitely do, and that's very commonly used. But so we are going to send you details about this. Briefly, so that's a very general aspect of the data we are using when looking at the site frequency spectrum. But as I was explaining in the course, there are other aspects of the data we could use to learn about the population demography. Very, very briefly, as if you followed my course, you understood that under each genome is actually a patchwork of genealogies, gene genealogies that are each of them a different realization of the same demographic history. So you could use directly these genealogies to learn about the population size changes. And it's actually more informative than the self-frequency spectrum because we looked at both mutations, self-frequency spectrum, and their frequencies, and also linkage disequilibrium between these mutations, haplotypes, how they correlate together in the populations. And this is also very informative about the demography. So if you combine both things, you can actually learn very precisely how population size has changed a long time. So that's what I'm showing in my course in one of those last slides where we see the population size with time in different populations. So it's not only that you can detect expansion, you can actually say what was the population size at this given time in the past.
Thank you, Etienne. I think there will be a follow-up for this question, so we can continue. Another question is, thank you for the information. I would like to know more about the origins and ancestry of Berber or Amazigh, if I pronounce correctly, Amazigh people from North Africa.
Yeah, sorry, I don't know a lot about that myself.
I could make some comments, although I haven't worked with on this region, so I'm not very perfectly, I'm not the right person to answer, but still, what I know is this ancestry is more closer to the Neolithic populations in the North Africa. So and we also know the Neolithization of this region was different than Europe. And they have an ancestry, if I'm not wrong, the ancestry that we find during the Neolithic is closer to the late Stone Age populations. And this ancestry today is only found in the northern North African populations, not in Europe, besides the admixture events. So my estimation would be, or suggestion would be that this Berber ancestry is something that we had in the region before the historical migrations, and which is probably connected to the Neolithization of this region. But I think
I confirm everything. This is also
okay.
what I know from that region. Yeah.
So that's all I can say. But
a lot of migrations.
If you have more questions,
yeah, sorry, a lot of migrations coming from the Near East, the Near East into North Africa changed the gene pool, but it seems like Berber and Amazigh were like more connected to the first populations who peopled this place.
Yeah,
during the
and this ancestry, it's connected to the earliest Near Easterns because where we define it as Natufians in like dating back to 12,000 years ago, and they're distantly related to this ancestry. But if I remember well, the Neolithic ancestry we found in the late Stone Age ancestry we find in the northern Africa is also connected to the distantly to Near Eastern ancestry. And if you have further questions about this, also we can continue in the discussion of the discussion form. And then I continue with the questions from Cécile. Have there been studies about Inuktitut ancestry? Okay, especially from Greenland. If I'm not mistaken, the question is still open regarding the datation of the population process for this remote territory. Any insight would be appreciated anyway.
Tricky one as well, very, very precise. So the peopling history of Greenland is characterized by the migration of, I think, if I'm not mistaken, 3 groups. The first is the initial peopling, is related to the initial peopling of the Americas. The second is by Paleo Eskimos, who did not contribute. This is the way they are called. even if Eskimos is a derogatory term now. So Paleo-Eskimos who did not contribute to the, or contribute very little to the current population. And then what we call Neo-Eskimos who derive from a third migration, actually relatively recently, but I don't want to get further because I don't want to say something wrong. I think it's in the last 4,000 or 5,000 years, I think. So that's relatively recent. And it seems like this is also disputed because some papers tend to conclude that there is actually a contribution of Paleo-Eskimos into the gene pool of current Greenlandic populations.
Thank you, Etienne. Another question from Sergio. Using
That's just a comment, actually, just to confirm.
sorry, yes,
yeah.
that's true.
Thanks, Sergio. But
Okay.
yeah, exactly. So if you look at other markers. And
I don't know if everyone sees the comment. I can just read the comment
sure.
in case. Yeah. Using YSTR data, there's about 1.2% variation with the Iberian populations indeed. Thanks. Okay. So the next question from Lucia. Why is your opinion about the use of the racist concept still being used, for example, when you submit a paper to scientific journals? They still ask you to clarify yourself in terms of race. Is that correct? Thanks.
Okay, so the term race is much more used in North America than in Europe for historical reasons, because it used to be also the way extreme right parties classified populations at some point in history. This is less a historical problem in North America. So that means that basically the term race has a strong historical connotation. At the biological level, we don't have a very clear definition of race, to be totally honest with you. We tend to use race in biology to designate groups of domesticated animals. So that means these are usually populations that are under strong artificial selection that is driven by human. And so they tend to have fixed genetic differences between these different races, if I use this term for example, dogs. So that means the mutation is 100% in one race and 0% in another one. So, but that's not a very well in biology in general. Not only for historical reasons, because we are still part of society, and we care about this, but also for biological reasons, because we tend to simply have the species level, which is very well defined. Species means a number like animals that or plants, living organisms that can have a fertile siblings, basically. And the term population also has a very strong and clear definition. Population means individuals that have the same, like, equal probability to intermarry together, what we call panmixia. So, and the intermediates between the two are extremely difficult to define, particularly in humans where they have this historical connotation. In any case, just to be extremely transparent about this, you can always classify individuals based on their genetic data. You can actually make the difference between two families, brother and sister. Actually, any individual except twins are any individual is genetically different from other individuals, so you can always make the difference between this individual and others. So the different levels of hierarchy we put and classification we put in these genetic differences is just a matter of interpretation. So as a short answer, I would tend to say no, it's not really correct. And the reason why it's still used a lot in North America is because it tends to give some proxy of the medical condition and the socioeconomic status where you are, which is not good, because these associations between race and socioeconomic status are or medical status is very relatively poor and weak, and it tends to reduce individuals and to their ancestry, which is definitely not correct. So we should go for what we call precision medicine, where medicine is tailored to each individual, rather than tailored to a race or population or so on. There is, as we explained in the course, a lot of diversity within populations. 80% of our genetic diversity is found within populations versus 20% between. So if there are, for example, genetic mutations responsible for disease, most of the differences will be between individuals and not between populations. So we should care about this and not races.
Thank you, Etienne, for the well-developed answer. Another question from Nick. How do you tell apart subsequent waves of ancient admixture? Example, 2 pulses of Denisovan admixture into the genome of a third population.
Good question, Nick.
Yeah.
So we have direct evidence of two pulse admixture from Denisovans in Papuans, and I'm going to explain how we have found evidence of this. Okay. So how can we prove that there is admixture between Denisovans and ancestors of Papuans in the first place without talking about 2 events? So the way we do that is to look at the genomes of present-day Papuan individuals. And we see places of the genome where we have a stretch of DNA with some mutations that are very characteristic of Denisovans and not of other humans, and in particular modern humans in this case. So we tend to say, okay, this segment of DNA was probably inherited from Denisovans in the ancestral population of Papuans. So that's a statistical method. I don't get into the details, but it works relatively well. This segment is characterized by 2 different things. Its length, how many kilobases the segment is, and also its level of genetic relationship with the Denisovan genome. These are 2 important parameters. The length of this segment tells us about when admixture occurred. The longer the segment, the more recent the admixture. The shorter the segment, the more ancient the admixture. First observation. The second, as I said, is at which extent the segment inherited in Papuans look like the Denisovan genome. And in this case, we can actually count the number of mutations between the Denisovan genome, the parental genome, if I can say, and the segment inherited in Papuans. And this also gives us a clue about the relationship between the Denisovan source population in Papuans and our Denisovan reference genome. And just to conclude with the answer, how can we know that there are 2 pulses and not 1? Because we would see 2 groups of segments, small ones and large ones, for example. So if we plot the distribution of the length of these segments, it's going to be bimodal, 2 different peaks, suggesting that there has been 2 events. And the same is true for the relationship between the segments introgressed observed in Papuans and the Denisovan genome. We also see a bimodal distribution, which is suggesting that there has been introgression from one group that was closely related to the Denisovan genome, and another group that is distantly related to the Denisovan genome.
Thank you, Etienne. We have like 10 minutes left, so I'm trying to be faster.
I'm going to do shorter answers.
And anyway, if the questions are left, then we will answer them in the discussion. It is already said. So from Gerald, can physical traits match with corresponding genes of some ethnic groups can be tracked down geographically with paleogenetics? Could some Latin American population be genetically related to Asian population through a potential past migration? So if I could start briefly, I say definitely there is, I mean, the best way to track down a physical trait, if like saying if it's belonged to a ethnic group, a historical group in the region, would be paleogenetics. But as I already explained, the how this comes to the point of fine mapping, like if it's like to finding the genetic characteristics connected to related to ancestry, local ancestry in the present day populations. However, one idea could be if you see the same variants that we detect in the present day populations as well as in their ancestors in the region, but not in the other from the same period of from other regions, then this could propose us the continuity of one genetic variant, like in the in particular geographical location. So could some Latin American population be genetically related to the Asian populations? If we talk about their Native American ancestry in the Latin American populations, yes, definitely. If you look at the most recent common ancestors of Asian populations and Native Americans, if the recent the most common recent most recent common ancestor carrying the same trait, then indeed this would be direct evidence of the migration events for the some traits. But what would you like to say, Etienne, about it?
So for the first question, can physical traits of some ethnic groups be tracked down geographically with paleogenomics? So what I understood from the question is, can we reconstruct physical traits from genomes? Is it the way you understood it or not?
What I understood is that the physical trait is really coming from a one ethnic group historically or not, like that's found in their ancestors or not. That's how I interpreted the question.
It's not an easy answer,
Yeah.
actually. But very briefly, just to complement what you said, Oguzhan, a physical trait is not caused by a single gene, as you say also, Girald, because you refer to genes. So you need to be to keep this in mind, really. So each trait, even skin pigmentation, which is relatively simple, is caused by dozens, if not hundreds of genes. Height is associated with more than 1,000 genes in the human genome. So that means that it's not that easy to infer or predict a phenotype from a genome. The first reason is this polygenicity, the fact that traits are very polygenic. And the second reason is that traits are not only caused by our genetic material, obviously. So the vast majority of traits have a given heritability, genetic heritability, meaning that 20, 30, 40, 50% of their variance is explained by genetics, but the rest is actually explained by the environment. So yeah, that means that a lot of our physical appearance, for example, is also driven not only by our genes, but also by our environment, including the environment, the developmental environment that we experienced. And about Native American, I have the same answer. So the divergence time between Latin American, if you get rid of the ancestries they receive from other populations like European and others, Latin Americans separated from East Asians around 30,000 years ago, which is not that far from the time when Native American, North and South Americas were peopled.
Thank you, Etienne. I continue with the questions. One question from Iman. Please, do you have any comments on the ancestry of Arab or more specifically Middle Eastern populations? In case of studying any of these populations, do you have any recommendation regarding reference panels that may be useful for global and local ancestry inference?
Yeah, we will. I have specific papers to share.
Okay,
yeah, definitely.
Which we can add in the discussion.
Yep.
And one question from Denise. I'm currently working on 𝐏𝐚𝐬𝐭𝐞𝐮𝐫, one of the hunter-gatherer populations here in Kenya. we are done with DNA extractions and now planning on genotyping using the H3 SNP chip. Exciting. Ideally, we could have sequenced the genomes, but funds and analytical skills failed us. What is your comment on the choice of type of SNP chip? Are there any plans to organize genomic data analysis courses for starters like us? Thank you for the good work.
Okay, exciting. 𝐏𝐚𝐬𝐭𝐞𝐮 are one of the few hunter-gatherer populations from East Africa. So it's very interesting to study these populations. I have the impression that they were also already studied, I mean, a few samples in previous papers that I can share with you, Denis, suggesting that they have the genetic ancestry close to their agriculturalist neighbors, but it's something that should be carefully checked. About the H3 SNP chip, I think it's a good idea to use that chip, which was specifically designed to study African populations. And given the fact that I'm expecting 𝐏𝐚𝐬𝐭𝐞𝐮 to be not very different from surrounding populations, you shouldn't have a big ascertainment bias for this data. Plan to organize genomic data courses. Thank you. So for Denis, if you can clarify what type of information you would like to get from these analysis courses, that would be very helpful. Because there are a number of courses, but it really depends what you would like to do. And yeah, more detailed ones. And please also clarify in your question if you would like this course to be online, or you would like to, you would be able to move to a new country, for example, or something like that. So that we could choose the one that fits the best your needs.
Thank you, Etienne. Maybe final question to answer from Kaina. If we take example the Malagasy people, they have an African ancestry and Austronesian with a kind of equal population proportions, if I'm not mistaken. How is it that the proportions are the same?
They are not the same. Within Madagascar, they vary quite a lot, actually. If my memory is correct, I think Austronesian ancestry is much more frequent in the heart of Madagascar compared to the coast, where we see much more African ancestry and actually Bantu related ancestry. So why is it half-half? I'm not sure. It's not half-half first, and it's quite different along within the island, the Madagascar island. And very briefly, I think it's these proportions are mostly driven by two basic parameters: how many individuals migrated. So if you have Austronesians came first, probably in Madagascar, and they were not that much actually. I don't think it's published yet, but colleagues of us estimated that the population that migrated to Madagascar was actually very small, from probably Borneo. And then the newcomers from Sub-Saharan Africa were probably numerous and maybe approximately the same population size as the ones of Austronesian people who were already there. So at the end, if you mix 50 with 50, you get 50%.
Thank you, Etienne. I think we are, Clémentine, I think we
Yes, I think it's time to close. And I would like to thank you, Etienne and Oguzhan. It was really a good session. I'd like to thank also all the learners. Thank you for your question, and don't hesitate to ask again your question in the forum, especially for the course, for example, it's possible to answer afterwards, okay? And don't forget about the exam as well. We'll post the exact timing as well. That's it for me, but do you want to have a final word?
Oh, I just wanted to thank everyone for the great questions. It was very cool. Thank you very much. a real pleasure to discuss about all these different topics. And we can already see that you all loved all these stories as we do. So thanks for sharing your interest.
Thank you, everyone, and bye-bye.
Thank you, everyone. Bye bye.